Okay, great. We're going to get started. So good evening, everyone. Thank you so much for joining today. We have some exciting talks lined up on the topic microfluidics for tissue-level transcriptomics, as well as a very exciting discussion and virtual networking sessions. So before we get started, for some of our newer audiences, I would like to introduce BAM, our group. So Bay Area Microfluidics Network organizes events to bring together leaders in microfluidics to foster innovation and collaboration. And currently we're a team of 5. So I am Anushka Gupta. I'm one of the members of BAM. I'm also a PhD student at UC Berkeley in the bioengineering department. So Thomas, if you can introduce yourself.
Everyone, I'm Thomas. I'm a 6th-year soon to graduate PhD candidate in Lydia Stone's lab and also in bioengineering at Berkeley.
Yeah, so we also have Adam and Samira. Adam is currently working at Apple and Samira is a hardware engineer at X, and they are both part of BAM as well. And then finally, Diego, if you could introduce yourself.
Hello, everyone. Thanks for coming. My name is Diego Heike. I'm a 4th-year PhD student in mechanical engineering at Stanford University. I'm in the Stanford Microfluidics lab, so my advisor is Juan Santiago. And actually, now that I'm here, I'd like to talk about this very exciting program that we've been thinking about within BAM. And this is the BAM Mentorship Program. Okay, so this program would be kind of a laid back, but its role would be to foster communication between those experts who have been in industry for several years and those with few years, or those are still in graduate school. Those would be our mentees. For those mentees, they can think of it as a casual conversations with a role model. And those conversations can take place 3 times a year. And we really want to encourage these conversations between mentors and mentees to the point that we are currently planning to provide Uber Eats or even Starbucks gift cards for each meeting. So you can sip a cup of coffee while you talk with your role model. And this is in the future. So we're thinking of starting in fall 2021. Most importantly, right now, we are looking for mentors and mentees to sign up. So you see that there is a link here below. I'm going to copy and paste it into the chat. And this is a non-committal sign-up sheet. So we just want to hear whether or not you would like to be a mentor or mentee and why you want to do it. So we hope that you seriously consider joining this program, and thank you.
Great. Thank you so much, Diego. So if you want to know more about BAM as a group, you can definitely go to our website, which is bayareamicrofluidicsnetwork.org. You can also reach us at .org, and we also have a LinkedIn group, which you are definitely welcome to join. So for tonight's program, we'll get started with our talks from Dr. Govind at around 4:45, followed by Dr. John, and then we'll have a discussion led by Dr. Don Arnold, and finally we'll start with our networking session at around 6 p.m. So a few guidelines for the event. Participants will remain muted until the virtual networking session starts around 6 p.m. Please change your Zoom name to reflect your first and last name, followed by your affiliation. For questions for our speakers or during discussion, please type your question in the chat and send it to everyone. We will be those questions will then be discussed during the discussion session by our moderators. And finally, for the networking session, we will be using Zoom breakout rooms. Most of you are already pre-assigned to the breakout rooms, depending on your interest during the Eventbrite confirmation. But otherwise, you would also be assigned based on interest. I would be in the main breakout room, so you could ask me, or you could also choose your own breakout rooms for networking. And so thank you so much. And then I'll give it back to Diego to introduce our speakers.
Great. And this next speaker, I'm very happy to introduce because he used to work in our laboratory. So our first speaker is Govind Kaigala. He is currently a staff scientist at IBM Research Europe, based in Zurich, Switzerland. He received his PhD from the University of Alberta and was a postdoc at Stanford University before joining IBM Research. At IBM, he leads activities on liquid scanning probes and their applications to biointerfaces, and a few years back initiated a program on next-generation tumor profiling systems that combine microscale assays and data analysis for precision medicine applications. He is passionate about translational, clinical/medical research, and with his team strives to bring quantification in biology and medicine by leveraging micro and nano systems. So Kaigala, the floor is yours.
Thank you very much, Diego, and thank you very much, Anushka. I'm really happy to be here. It's a little bit of an odd hour in Zurich, but hopefully I'm not blabbering odd things. But I think there was a time I used to be while I was in Vaughn's lab, there were days where I was doing experiments, or let's say there were nights when we were doing experiments at this hour too. So it's a few years back, but still I do remember working at these hours. So let me start by sharing my screen. So really happy to be sharing our work with this forum. And unfortunately, this forum wasn't there at the time when I was in the Bay Area, but happy this exists. And okay, sorry. So I'll be talking about two aspects. One is about a technology called microfluidic probe, or I'll give, or what we also call as open space microfluidics technology. And the second aspect is how we would be using it for spatial transcriptomics. Actually, we didn't set out to work on spatial transcriptomics per se, but this was along the way how we got into this particular aspect and have been working on it. So I'll give you the motivation of why we were working on this. But before that, actually, I was speaking to Don earlier this evening, and it came across that it could be interesting to give a bit of a perspective of where I am and in terms of this institute, and what is the larger role our group plays in this. So our team works largely on compartmentalization and microscale molecular assays, and we try to see how we can use these to develop data-centric bioanalysis. And much of the technologies that we are working on goes in the direction of personalized medicine. So this work goes on at the IBM Research Lab here in Zurich. The photograph in the left is our campus. There are about 350 scientists working out here, including PhDs and postdocs too, about 80 of them. And the right is the Binning and the Rohrer Nanotechnology Center, which is a joint center between ETH Zurich and IBM Research Campus, based on our campus here. So my team has expertise basically in microfluidics and micro and nano fabrication. Also, I would say we have a very good biology and pathology. And the common theme which connects these two, I would say in some ways, is chemical engineering, not necessarily to scale up aspects, but to actually scale down. I don't need to explain this to this audience because I would imagine all of you are really very familiar with what we really scale down in microfluidics. So I organized the talk into a few parts. Briefly to give you the idea of why we are working on this topic, it's in the context of measuring and creating new metrics for heterogeneity in cancers. I'll talk a little bit about the microfluidic probe technology and how we are applying to spatial transcriptomics, and maybe just give a bit of an outline, time permitting, on how we are getting to multi-omic profiles and how spatial proteomics and genomics also has a role to play in it. So heterogeneity in tumors is really a very complex topic, heterogeneity is really present at so many different scales and almost at all the omic levels in cancer. This is a figure that we created for a review. We are in advanced stages of writing currently. So we are very interested in looking at intra-tumor, inter-patient, as well as intra-patient heterogeneity. That's the area that we focus on. But really, there is a lot of heterogeneity present at the spatial level. There is heterogeneity present at the temporal level. So how do we capture spatial as well as temporal heterogeneity is the key objective. Of course, there are many other aspects of intrinsic heterogeneity as well as extrinsic heterogeneity. We look at certain aspects of those, but our focus is right here. To this end, of course, we need to look at heterogeneity in different omic levels. We're looking at some of the proteomic level, we're looking at it at the genomic level, we're looking at it at the transcriptomic level, and of course, at the chromosomal level too. So when we started looking at that, we realized that we need to start developing new assays, which is compatible to trying to extract information at this multi-omic level. And also we started asking ourselves this question that if the heterogeneity is present at the spatial and the temporal level, we may need to start doing some form of compartmentalization or some form of localization to be able to perform all these assays together. Along the way, we started looking, coincidentally, we realized that there is a lot of innovation which is happening in the spatial transcriptomics field. Be it using barcoded approaches, using beads as one example, or using the many interesting variations of fish-based methods. I won't get into the details of all of these. This is just to show that there is lots of active work going on this. In fact, last year, or just late last year, there was a Nature called Spatially Resolved Transcriptomics as the Method of the Year 2020, and it was on the cover page. So just to say how important and how recent this work is, there are times that we work on topics, we are always doing catching up. But this was one of the times we found it quite interesting that while we were working on this topic, we saw that there were lots of interesting papers being published and also being named really the paper of the year or the method of the year. So as mentioned, that to understand heterogeneity, we need to do compartmentalization of assays. So typically in molecular biology or conventional biochemistry, which is applied to diagnostics, cell biology, drug screening, or many, many other fields, there is one reagent which is applied to one sample, and you do you ask one type of question and you get one type of answer. That's typically, this is an abstract view of this, conventionally what happens. Our view was, I'll put this specifically in the context of tissue section soon, but can we, within one of the sample, perform different forms of compartmentalization or localization to perform all these different colored assays, let's say. And so we put this idea in the context of tissue sections. So typically when a diagnostic or a cancer type test is done, in addition to imaging and looking at other markers, for solid tumors particularly, it's very common to take a biopsy sample, make a FFPE section, and then be able to form this type of sample. From there on, as I showed in the previous animation, one sample, one test, one potential answer, or one potential question is asked and one potential answer you get. That's what you see here. So it comes down to how you budget the sample to perform different tests. With that, of course, what we currently have is potentially a partial profile of a tumor. Our thesis or hypothesis a few years back was potentially you could take one tissue section, perform many different assays on top of it, each of them adjacent to the other. Of course, there are certain guidelines, rules, algorithms which we have come up and we are developing right now to be able to, what is the optimal sample, what is the minimum size of the sample, and how they should be spatially resolved such that you're getting an optimal map of the information that you're trying to procure from each of the sections. And you can do this type of mapping differently on consecutive sections. With this, we think we will get a very different type of profile, a 3D profile eventually of a tumor, and potentially a more complete profile. With that, of course, we can potentially perform more personalized therapies. So again, a very artistic, non-technical view of this is a biopsy sample, you section, one test, you look at, maybe you ask some question of genomics, certain mutations you're asking about, then you ask about certain state of a chromosome, whether there's a particular translocation, or you're looking at a particular protein expression, or the most simplest thing of you're looking at morphology. This is the standard procedure. and we talk about something called tissue microprocessing. Can we microprocess a tissue to perform all of these tests next to each other? For that, of course, you need some compartmentalization techniques. And compartmentalization techniques, which is compatible to biological samples. And that was the genesis in some ways of one of the techniques, what we call the microfluidic probe. But this falls under a broader family of open space microfluidics, where you don't take a biological sample from its more or less native state and put it into closed microfluidic channels, but rather bring a microfluidic system or a network onto these biological systems, which is where it's more native. So in some ways, it's really a very simple, almost trivial system what we have. Think of it as two pipettes. One pipette to bring the liquid in, the red colored liquid to process the surface, and then adjacent pipette, which is taking out some of the liquid. There are certain design guidelines. You take out more liquid so that you are hydrodynamically focusing the red colored liquid that you're injecting. Of course, now it's not in the context of pipettes, but it's a microfabricated device where you're having two channels in the base state and you're injecting red colored liquid, you're aspirating much more than what you're injecting. And the biological sample is typically placed on this buffer put on it or some sort of cell culture media or so. And that liquid, the blue liquid, is hydrodynamically focusing the red liquid. So when you see through an inverted microscope from bottom up, you will see this type of teardrop shape. This is a function of where the two apertures are placed. But depending on how you place the aperture, you could shape the liquid in completely different ways. So we have a complete, a very, very large family of devices to do these type of hydrodynamic flow focusing at the tip of these probes. So these probes are mounted on XYZ stages. A biological sample is put here as an example, a microtiter plate, a glass slide, a petri dish, and they're imaged from the bottom. And so you have spatial and temporal control of where on this sample you're able to perform which type of reaction. By the way, this is a simple prototype device that we built some time back. We have a large family of these also. Depending on the type of application, we try to build a prototype which looks almost like a product, but I must stress it is a prototype to be able to do good repeatable experiments. Again, this is just one more animation of the idea of how we do hydrodynamic flow confinements in an immersed substrate. There are many, many variations of these type of devices, as I mentioned. So this is a very simple hydrodynamic flow confinement. You could have a hierarchy of these. You could have like Mashushka dolls almost. You could have one liquid confined inside another. You could have radial shapes. It's actually very interesting. It's almost how much creative you want to be. You could have that many different types of shapes. We modeled the physics of this, of course, to be able to predict the shapes before we start designing. We have very interesting, simple models actually for this. This is a case where you have 4 different apertures. You're injecting the liquid in the middle, the blue colored liquid. The outer liquid is helping focus this, and there's immersion liquid all around it. Broadly, you can classify these in 2 ways. One is from a functional standpoint. One is to bring something onto a substrate, or let's call it a tissue section henceforth, where you want to do some sort of a deposition. You could perform FISH, you could perform RNA-ish, you could perform immunohistochemistry. In the right, what you see is you can take away samples also. The same liquid which is bringing something, or another liquid which is bringing something else, could be a starting point for, say, disrupting a tissue and being able to take its contents. That's what you see in the right. So we have been working on quite a bit on both of these approaches. Both have different applications for this idea called tissue microprocessing. Some time back, we started working on microfish. We had the opportunity, really, we asked ourselves, can we go back and look how the world of fish has evolved over a period of time? And we took the liberty of calling the recent implementations of fish modern fish, in which there is a lot of innovation in the probe design, going from very standard probes, DNA probes, which are there originally, to, I would say, synthetic PNA type probes. And really, there's a lot of innovation out there. One of the recent, from a technology perspective, actually, there isn't much. So one of the implementations, or few implementations, has been on microfluidic fish. And what we bring to the table is more localized implementation of fish, which is here. Again, I don't have to preach to the choir here. What microfluidics brings to the table in this is the ability to be able to reduce the consumption of the fish probes. What this particular type of microfluidic system, which is the MFP or the open space microfluidics brings is the ability to localize, the ability to perform fish. And I wanted to emphasize this particular point here. Because you're having a continuous flow of the probes, fish probes on top of the tissue section, you're having a constant flux. Because you're having a constant flux, the concentration of the probes is constant in the vicinity of the cells which are permeated. And because of that, the hybridization time is not dependent purely on diffusion to bring the probes. We are having a much faster hybridization time as compared to pipetting because of the MFP. And I won't go into the details here, but we're getting a very nice, beautiful signals, fish signals, in at least 10 to 50-fold reduced timescales compared to conventional pipetting. We also developed along the way, incidentally, something called real-time fish, analogous to real-time PCR, where you're able to see the evolution of the fish signal over a period of time. Again, what you're seeing in this movie in the right is where you're bringing probes, then you're bringing a wash liquid, you're bringing probes, you're bringing a wash liquid. So you're able to see the evolution of the signal. With this, we started being able to see how we could potentially think of doing a rational design of a fish assay, which currently does not happen. Sorry, I'm skipping the details because I just wanted to give, in this time frame, the big picture of how an interesting, newer, different technology can have an impact in a field, which is, as an example here, RNA, as an example, transcriptomics. So we started working on then spatial transcript expression also. We did something similar. We did pre-treatment, we got the probe to bring the RNA probes, and then we were able to do signal amplification. With this, we could again start exploring the expression of HER2 in this particular case across breast carcinoma tissue sections. We could look at expression in different parts of the tissue section, and we could see, again, not getting into the details, but you could see quite a bit of variation of the expression states. But often, spatial transcriptomics has to be complemented with other technologies to evaluate the profile of a tumor. That's exactly, we feel, one of the limitations of currently some of the state-of-the-art transcriptomic techniques. They're difficult to complement with other techniques. But since we're using the same probe technology for a bunch of different approaches, in this case for even performing local immunohistochemistry, it was possible to compare and perform all these different tests on the same section. This is an approach of how you perform local immunohistochemistry on a tissue section. Typically, immunohistochemistry is to measure the protein expression. It's a very qualitative approach, which has been implemented for tens of years now in the pathology community. We asked ourselves this question: why do pathologists look at only endpoint signals? Because that is what it is. We realized that there is no good way to look at evolution of the signal when you're not using fluorescence. So we came up with an approach of how you could sort of have a pseudo kinetics curve that you would create. Because we're doing localization, we could perform different tests with different incubation times, one next to the other. And because there's a continuous flow, the rate at which this reaction happens is much faster. With that, we sort of create a pseudo kinetics curve. We have a database which we created for different expression levels of different cell lines. With that, we were able to find a sort of an approximate level of the expression of a particular protein and be able to give a relatively more quantitative type of profile for the protein expression.
Hey,
Then, of course,
just to let you know, we're going to move on to the next presentation in 3 minutes. Just as a heads
absolutely.
up.
Thank you. I'm getting closer to the end of it. So we were able to perform the multi-omic aspect by integrating both the transcriptomic approach of the RNA-ish, I talked about 2 slides back, and the proteomic approach here, and do a very nice comparison to look at spatial heterogeneity. Lastly, we also started looking at the genomic analysis, not just the transcriptomic for heterogeneity. We were using the same approach of being able to extract different parts of the tissue section, and we were able to connect these samples to perform in the workflows for doing sequencing. We had certain challenges of purification of the sample, and then electrokinetics was, of course, to the rescue, and more specifically ITP. I was quite happy for that because we did quite a bit of ITP assays back in the day, and we were able to use that readily to purify the samples and concentrate it before we could put it in the workflow for sequencing and look at certain SNPs in this case. We were able to do something very similar again with protein expression and be able to look at heterogeneity. So essentially, we found 4 different ways of trying to start mapping heterogeneity. And we are actually at this state that we have interesting assays to be able to look at the genomic level, the proteomic level, the cellular level, as well as just the histology also. Now we are starting to see how we can spatially combine all of this data to get some sort of semblance of complete profiles of a patient sample. And of course, a very interesting idea or a concept here is what we're doing right now is all measurements. And all of you are very aware that many in microfluidics community is trying to take quantitative ideas and try to make it, qualitative measurements and try to make it as quantitative as possible. But measurement is not the end point. Trying to transpose something from a measurement to what would be useful for a clinic is a completely different ball game. And that's exactly what we're working on right now. We're working with many clinicians to see how we can translate this aspect. So with that, I would like to stop, acknowledge my colleagues, students, the team, collaborators, and happy to take questions now or later during the discussion.
Thanks, Kaigala. Actually, we'll take questions during the discussion, but if you have a question that you want to ask, you can type it into the chat now. And when Don is moderating the discussion later, he'll bring up those questions. So make sure to type them into the chat if they pop up. Thanks, Kaigala. So now we'll move on to our next speaker. Our next speaker is John Bashkin. He is a co-founder and VP of Business Development for S2 Genomics. He has 30 years of experience leading innovation programs and business development in the life sciences. He previously held senior positions at Molecular Dynamics, where he leads the development of the Mega BACE 1000 96 capillary sequencing system, Amersham, and SRI International. He received his PhD in chemistry from Indiana University and an MBA from Santa Clara University. Bashkin, you have the floor.
Thank you so much. Time check, I have 15 minutes for my talk, is that correct?
That's correct.
Okay,
yep.
Great. Let me share my screen. Thanks everyone for coming today. I'm going to tell you about what we're doing at S2 Genomics. It's not actually a microfluidic device, but hopefully you'll find it of interest. We're in the space of single cell biology. It's not spatial transcriptomics, but it is single cell genomics. And this is an area that's really exploded over the last 7 or 8 years and has come to dominate not only genomics, but really most of biology research and a good portion of translational research now. The reason it's so exciting to be involved in single-cell genomics these days is it's really having a tremendous impact on our understanding of biology, looking at the heterogeneity inside of tissue that wasn't appreciated before. Cells that are supposedly exactly the same type, in fact, can behave quite differently depending on their environment. New types of cells are being discovered. The biology underlying cancer is being taken to an entirely new level of sophistication, complexity, and understanding. And as I mentioned, all of this work is now having a direct impact on clinical research and clinical applications. So consequently, from an economic standpoint, from a business point of view, this area is huge now. It's a little under $3 billion last year in 2020. The company that dominates this area is 10x Genomics, and their valuation is somewhere around 20 billion now, and they're only a few years old. So it's really taken the biology world by storm. But there's a problem. So this is the workflow for doing single-cell analyses or single-cell genomics analyses if you want to start with solid tissue. And so typically, you'll end up with a suspension of singulated cells or nuclei. And those then get fed into a system sold by 10x or a system sold by Dolomite or one of the other competitors. And that generates your sequencing library on a cell-by-cell basis, and then they get analyzed on a sequencing system from Illumina typically. So everything from this middle arrow of having your single cells or nuclei downstream is very robust now. The experiments are complicated, but the systems are very high performing. The throughput is tremendous. You can analyze tens of thousands of cells in any given experiment. But 10x has said, publicly on multiple occasions, that the biggest problem that their customers have is upstream. And that is the ability to take solid tissue and dissociate it into the individual cells or nuclei that make up that tissue and create a purified suspension. And if you're doing cells, you need to do it in such a way that your cells are still viable, they're still happy, you haven't induced cell stress-related genes that will affect the transcriptome you're trying to measure. So it's a very challenging problem. And in genomics, by and large, these dissociations are done by hand. It requires a lot of skill to do it. We've been told by investigators that it can take anywhere from 3 months to over a year for their postdocs, technicians, whoever, to learn how to do this and do it well. There are not standardized protocols, so you can have 2 different labs working on exactly the same tissue and get different results. The isolation itself can impact the transcriptome that you're trying to measure, as I mentioned, through induction of cell stress-related genes. And you can lose very fragile cell types during the process. And so the representation of cell types in your analysis may not reflect what was in the original sample. So you have a bias there. If you want to look at nuclei rather than cells, and there are a number of reasons for doing that, then again, it's time consuming, it requires a lot of skill, and you have to be very careful not to compromise the structural integrity of the nuclei that you're trying to isolate. So the result is you have data across labs that can't be compared. You have artifacts built into your data from the dissociation process that you really can't take out in bioinformatics because they're built in and you don't know really what they are. So we set out to address this issue by introducing automation. I'm sorry, but before I mention that, the reason why this is such a huge issue, it's not just a niche problem it's not just a niche issue for 10x genomics users, but this is a problem that goes all across biology research now. For single cell genomics, for flow cytometry, if you want to isolate immune infiltrating lymphocytes, tumor infiltrating lymphocytes for immuno-oncology, if you're in applied markets like ag bio, all of these different huge areas of biology all start with solid tissues that need to be dissociated at some point during the life history of those samples. So we introduce automation. Our first product is called the Singulator 100. It operates on one sample at a time. And we design it to make the tissue dissociation process incredibly simple to do. We have a single-use disposable cartridge with a cap. You lift this cap off, drop your tissue in, put it into the instrument, select your protocol, and say go. And that's it. If you're doing nuclei isolations, they take 5 minutes. If you're doing cell isolations, it takes between 20 minutes and an hour, depending on the tissue type. The protocols are all tissue type dependent and tissue type specific for isolating cells. The cost is modest. It's $35 a sample for the cartridge and reagents, and the benchtop system is $37.5. So it's very affordable and is really not just a nice to have, but really enabling for a lot of customers that we have for whom manual processing simply does not work at all for their tissue types. I'll show you a video later on of it in operation so you see how easy it is to use. These are the kind of results that customers are interested in getting. If you're isolating cells, you want the cells to come out with really high viabilities, preferably over 90%. We release a protocol when we get at least 70% viable cells. Usually we're in the low 80s to low 90%. In terms of the number of cells that you isolate, that's tissue dependent because every tissue has a different cell density. And so in general, for cell isolations, we're getting between around 20,000 cells and 100,000 cells per milligram of tissue. So if you throw in 10 milligrams of tissue, you've got hundreds of thousands of cells coming out, and what you typically load on a 10x system is 10,000 cells. So you need a very, very small amount of tissue to generate enough cells for your downstream analyses. If you're doing nuclei, we tend to isolate more nuclei than we do cells, and so here you're getting around 50,000 to a few 100,000 nuclei, again, per milligram of tissue. So you really don't need much material. I mentioned that cell isolations require different protocols, different enzyme cocktails for each different tissue type. But for nuclei, it's the same for everything. We really like working with nuclei, and probably 85 to 90% of our customers isolate nuclei alone, or they'll do a mix of nuclei and cells. There are so many reasons for logistical reasons and experimental reasons for working with nuclei. It's the same process for nuclei, like prostate tumors or hemangioma tissue. These are tissue types that for the customers just fail routinely if they try to do things manually. These are what nuclei look like, for those of you that may not have seen them before, and this is one of the key QC metrics that customers will use to evaluate the quality of the product. So these are nuclei from a variety of tissue types that have been stained with DAPI. And you look at them under a fluorescence microscope, and what people really want to see are these bright spots, because that's the staining of the DAPI dye on the heterochromatin inside the nucleus, inside the cell, or which came from the cell. And having intact heterochromatin is really key for successful experiments with single nuclei sequencing, in particular an assay called the TAC-seq. So that's one of the the key quality metrics that people will look for. These are data from the Riken Institute in Japan. And when they saw these and saw the intact heterochromatin, I mean, they're just head over heels happy with how beautiful these nuclei look. And you can see the different shapes, right? The heart nuclei, muscle nuclei tend to be elongated, whereas the liver and the brain tend to be very circular. So how does this type of system impact your data at the end? This is a data slide I got recently from a customer at UCSF, Heiko Yang, who's in Franklin Wong's lab. They were running human bladder, and they do this routinely. They have a manual protocol that they've been using for a long time on tumors. We'd never tried human bladder tumor before, and so this is their first try. The same size sample going into the Singulator or going into their manual protocol. And you can see the difference in the results. It's pretty dramatic, almost twice the number of cells. And then once you load onto the 10x sequencing array, a significant increase in the cells that are captured successfully, the number that passed their QC metrics, the overall yield, and all of the other metrics associated with doing a single cell or single nuclei sequencing experiment, the end feature, the end count, the mitochondrial contamination, and the ribosomal contamination were all significantly better with the sample processed on the Singulator. And if you're spending between $2,000 and $3,000, or maybe up to $5,000 on your downstream sequencing experiment, between the 10x cost and the Illumina cost, having this kind of improvement in the quality of your data is really significant. So I'm going to show you a video of how it works right now. The instrument that you'll see is a prototype in the lab, so it doesn't have the skins on it. But this is for a kidney, nuclei isolation from mouse kidney. Just lift off the cap. You can just drop a whole kidney in. That's a big sample for us, but it's easy. Select your protocol. You can enter notes about your run and saved in a log. Slide the cartridge in. There's a camera inside that shows you the dissociation in real time. So here we are adding our isolation reagents. And then there's a built-in spring-loaded plunger mechanism built into that cartridge cap. It's going to get depressed and spun around very slowly, providing very low shear on the tissue and disrupting that tissue mechanically and isolating the nuclei from the tissue. If you're doing cells, then exactly how that process works is a bit different. We need to be much more gentle on the tissue for isolating cells. But for nuclei, you want to go as fast as possible and just bust open the tissue. Then we pull a vacuum on it. There are filters built into the cartridge, so all of the debris, or most of the debris gets filtered out. Now we do a rinse and pull that through, make sure we get full recovery of all the material. And then when you're done, the motors will retract, release that that spindle, puncture a seal with your pipette out your suspension, in this case of nuclei, spin it down for 5 minutes, and there's your pellet of nuclei. And then you can go on with your single nuclei sequencing experiment. So the automation really makes life so much easier for people in the lab. Protocols that could take 2 to 6 hours are now 5 minutes to an hour, depending on what they're doing. It doesn't matter who's doing it. There's no, good hands, bad hands problem in the lab. Everybody gets exactly the same results. You can customize the protocols. You can use your own reagents. You have access to tissues that were not amenable to manual processing. You have access to frozen tissues for isolating nuclei that were not accessible before. And you can share protocols, load them onto a different instrument. So if you have a collaborator in a different lab, you can be assured that everybody is doing their cell and nuclei isolations exactly the same. So with that, thank you very much for your attention. we are hiring. I know there's a jobs board portion of this, so we have open positions for mechanical engineers, field application specialists, salespeople, and also a director of marketing position. If anybody either on the call or you know of anybody through your network who might be interested in joining us, we're in Livermore, California, and we'd love to hear from them. So thanks very much for your attention, and happy to take questions during the Q&A session.
Thank you for the presentation, John. And so now briefly, now we're going to go into the discussion part of tonight's event. And before we go into the discussion, I'd like to introduce our moderator, Don Arnold, who's been working with for a long time. Don Arnold received chemistry degrees from Cornell and UC Berkeley. He began his microfluidic career in 1997 as a senior member of technical staff at Sandia National Laboratories. In May 2000, Don co-founded Exigent Technologies to develop and commercialize electrokinetic and microfluidic systems for the life sciences. Exigent was acquired by SciEx/Danaher in 2010. During his 17 years with Exigent and SciEx, Don held several leadership roles that included responsibility for technology and product development, intellectual property management, open innovation, business development, and general management. In 2017, Don founded Veristad, where he provides technical and business consulting for life science companies, with an emphasis on microfluidics. Since mid-2020, Don has also been an executive in residence with General Inception, an institutional co-founder for science-based companies. So with that, I give the floor to you, Don.
Okay, I guess I'll turn on the microphone and camera here. And first start just by thanking everybody for coming out this evening, and especially thanking John and Govind for the great presentations tonight, coming across multiple time zones. One of the benefits for the Bay Area microfluidics meetings, maybe less beneficial for our folks in Europe who are taking time to make presentations for us, so thanks for that. Let's see, I think I want to start off a little bit. I try to cover some different directions of questions in a sense, and start off with a technical question that popped up in my mind for Govind, and I may have missed it, so I apologize if I'm asking a question you answered. But with I love the sampling concept and being able to introduce reagents as you're moving through the chip at the interface of the sample. What is the size that what's the smallest size that you've been able to demonstrate that you can actually map onto? And what's the limitation there?
Thanks, Don. So it's a valid question, and actually I did not cover it. So it's defined by a couple of things, but let's start with nominally what we do, and then I could say maybe how much we could push it in terms of resolution. So nominally we confine the liquid down to approximately, say, a spot of about 30 to 40 microns diameter. We could certainly go smaller. It's a function of how small we the channels, the apertures basically. We could make them, currently it's about 20 microns by 20 microns. We can absolutely certainly go down to a few microns, but there are a few challenges there. Not necessarily from a fabrication standpoint, it's more from a very practical usability standpoint. The resistance, hydrodynamic resistance significantly increases, and you need to start really filtering all the liquids. You need to start working under a hood. You need to be able to, it takes time to flush the system and have a clean system to purge the system. So from a very usability standpoint, we think having relatively large apertures is good. So one way we get about 20, 30 microns footprints, but you could also push that resolution without pushing the resolution of the apertures by actually focusing the liquid a bit more. So if you're having the shaping liquid, which is having slightly different properties, that could push down the resolution of the liquid that you're of interest.
Yeah, that's what I was wondering, because it can do a lot of cool things with confining your flow and controlling it. But that requires just a quick follow-up. It seems in all cases requires some pretty good flow control. And my background there, and always thinking about flow control to get the fluid in and out and maintain. How do you go about that with the systems that you're using?
So actually, yes, in principle, we started with really very sophisticated pumps and pumping systems, which are rather expensive because you need to be able to have low flow rates and which is not pulsating. But soon enough, we realized that that's one way to go. But there's one more way is just to be able to have a pressure difference.
Yeah.
So you could create that really by using pressure systems. And it's much more convenient doing that because it's much more scalable. But there could be a cross talk between different channels if you're using the same pressure system, but then it gets into the details of engineering. You can be able to minimize those. There are several ways. So over the period of time, we have found really very interesting, simple solutions to decouple the crosstalk, as well as to make it much more stable, and also to have a continuous feedback because you want to have some sort of a feedback, not just an open loop system.
Yeah.
And also one way, of course, because of my background a little bit in electrokinetics from the past, is we also showed ways of how we could have electrokinetic electroosmotic flow. And relatively simple, not much is needed. Put two electrodes and start moving the liquid.
Yeah,
so we demonstrated the same concept with just electroosmotic flow and gravity. And we have a variant of this called electrokinetic scanning probes.
Oh, cool. Okay. Well, that's really, if anybody knows my background, I'm a big fan of that one. So that's cool. For question for John, I wanted to give you a chance. There was and I'll check with Diego and make sure it wasn't just me. I had a little bit of dropout when John was just differentiating between cells and nuclei. Was that for everybody or just for me?
No, it's for everybody.
Yeah, maybe John, you dropped out a little bit during the cell nuclei differentiation on the platform. I was going to say maybe if you're willing to kind of cover that slide again real quick, it'd be good for everybody just to know, because you moved on to a different thing right after that. Would that be okay?
Yeah, absolutely. Let me just bring it up again real quickly.
Yeah.
In general, while the slides, there it is. Let me share again. So in general, the nuclei isolations are very fast, and we use the same reagents regardless of the tissue or the sample type. We have a standard protocol is about 5 minutes. For some tissues that are mechanically really tough, like prostate tumors can be mechanically very hard. So we have an extended protocol that takes about 10 minutes for those tissues to increase the yield of nuclei from those very mechanically hard tissues. For cells, those protocols are 20 minutes to an hour, because the first step when you do cell isolations from tissue is you first incubate the tissue with a mix of enzymes. For nuclei isolations, it's a buffer detergent that's just breaking open the cells to get the nuclei out. But for cell isolations, you use a mix of enzymes, and so the tissue is going to get incubated in the cartridge with that enzyme formulation for 15 minutes, 45 minutes, whatever the tissue requirement is. And then following that initial enzymatic part of the dissociation, then the mechanical dissociation comes in to finish it off and complete that. So that's the reason for the different protocols and the different time frames for the isolations of cells from tissues.
Actually, it was the slide. There was a slide a little bit lower that talked about the nuclei as the one where we lost you a bit. So, but that was new information on that slide, so that was good.
Okay.
Let's see.
This one.
Let's see. Help me out, Diego.
I think it was no, it's in slide 7. It was
Slide 7.
transitioning between
There we go.
slide
Yeah,
7
there we go.
and-
This one?
There we go. Yeah.
Okay, yeah, so I didn't bring up
Take that one.
the, I didn't include the slide that shows the tissues that we've done for cell isolations.
Okay.
It's a smaller list because most of our customers are doing nuclei rather than
Got it.
cells. But still, you know, it's a range of normal and tumor tissues, but just not quite as extensive. A lot of our customers really like working with nuclei for a couple of important logistical reasons. One is that a lot of the translational research going on is accessing tissues from frozen tissue biobanks. So institutions, they'll have tissue biobanks that might have, you know, hundreds of thousands of samples in them. And you can only isolate nuclei from frozen tissue. You can't isolate cells. And so if they're going to do research on tissues from those biobanks, then they have to do nuclei.
Okay.
If they want to do research on contemporaneous samples, they're getting tissue from an operating room, then the logistics can often be very difficult. So if there's a colon resection, it can take sometimes hours for the tissue to make it from the operating room to the lab where they're going to do the experiment. And the tissue's dying during that whole time. So by the time you get it to the lab, there's so much necrosis going on that it's very difficult to isolate viable cells from that tissue. And so what some labs are doing instead is they're just flash freezing the tissue in the OR. Yeah. And then they can analyze it whenever they want to, but they analyze it using nuclei rather than cells.
Yeah, so that really goes to some of the point that Govind was making around the spatial versus temporal evolution of a compilation of cells and being able to get an accurate assessment of the transcriptome at the point in time that you're really interested in seeing it. So it sounds like, you know, and I remember seeing this as really early on before, just before TCAT was set up, having a discussion with Chris Austin about this. And I remember a researcher asking the question, how can I get it so that I get my sample out and it doesn't change in time before I do my proteomics analysis? And that was a challenge back then, and I think it still is today, and being able to get there. So I think when we were talking earlier, you said they basically end up flash freezing right away after surgery or something, right?
That's right. I mean, not everybody's doing that, but a lot of labs are doing that. And many of our customers started their single cell work using cells, and now they've migrated to nuclei. So the whole field has really shifted dramatically to single nuclei sequencing, and people are getting over the sort of conceptual barrier of or validating that the transcriptome information they get from looking at just the nucleus versus the whole cytoplasm, with very few exceptions, the information you get is the same. There are, you know, a few genes that are going to be noticeably different in their expression. You're going to have long non-coding RNAs inside the nucleus that you won't see in the cytoplasm, but those you can account for in bioinformatics. So if you're for most experiments, not for everything, but for most experiments, you can often use nuclei instead of cells. Okay.
It kind of leads me to a question, you know, kind of for both of you, because you're going at it in different ways, and you've got both some pretty new technology with different subtleties around how they work. You know, would you say with the work that you're looking at forward, what's the biggest barrier in a sense at this point? Is it going to be the question of the hardware, or is it, you know, or is it going to be in terms of chemistries required to make it work, or are you at the point where you're starting to push the boundaries and get into data analysis and data mining types of problems for the customers? I mean, where do you see, giving us a picture of current status and where things are going from here for each of your technologies?
Govind, do you want to go first?
You're already at it. Maybe you start and I join you.
Okay. So for us, it's really two directions. One is continuing to increase the throughput on the system. So, you know, a 10x will have has 8 channels on it, and so customers like to be able to process nominally 8 samples in parallel, and so increasing the sample throughput is an obvious direction, but then also integrating more aspects of downstream assay processing or assay steps into the cartridge, further integrating workflows, introducing magnetic bead handling, so now we can isolate specific cell types within the cartridge, and create those conveniences and that automation integration for customers further downstream. Those are the 2 directions that we're pushing on.
Okay.
So I would say one aspect is common, what John mentioned for us too, is the throughput, because much of what I showed is, I would say, currently a little bit more in the realm of research. Although we keep in mind at all times what happens in the workflow. We work with a couple of sequencing centers and as well as with the clinicians too, to understand, or sorry, the researchers who are working in a clinic to understand what aspects of the workflow could be changed, or what aspects they will absolutely not agree to change. So we're trying to navigate that path at this point of time. The logical next step will of course be the throughput. And we also see the second aspect and a third aspect actually. A second aspect is how do we best integrate the multi-omic approach, which we are taking. And the third aspect from a more systems level perspective. And the third aspect is, of course, the data aspect of combining all of this. It's opened up a completely new area for us personally. There are people who work on it, but I think it provides data in a slightly different format. So we are trying to see how to best integrate that. There are lots of interesting mathematical approaches around it, and we're really currently exploring that.
Okay, cool. That's great. So there's a lot of, you won't be retiring anytime soon out of this. In either case, that's good. Let's see. I'll go to a couple of questions that are up on line here in the chat room. So from Hangwang Shu, for Govind, with continuous perfusion suction for deposition, is there a recycling system so that you get to some expensive reagents, you can cut your cost a bit?
Absolutely, absolutely. This was because it's a continuous flow. It's always almost the bane of existence of the advantage of flows. We would like to have flows, but at the same time, flows are not so conservative of the reagents also. However small the flow rates are, however small the volumes are. So we do have a recirculation system. It's challenging really to implement a recirculation system within microfluidic systems, but in a very simple way, I could explain it. So basically, in the chips itself, or in the heads of in the device that I showed, we have a reservoir in it, very near to the apex or the tip, which almost holds about 1 microliter volume. So we constantly switch the direction of the flows. So by constantly switching the direction of the flows, after say the 1 microliter has been used, so we are able to take the liquid back and forth. With that, what we did, we created some models of what is the consumption rate. So we changed the flow rate accordingly, such that more or less the reaction rate on the surface, which the liquid comes down, is similar.
Yeah.
So as the cycle number goes from say 1 to 10, the flow rate will appropriately reduce, so that the interaction time increases.
Okay.
So we do have the
so you
short
take into
answer
account the kinetics so that you continue to get
exactly.
a consistent response effectively. Yeah. Well, that's a clever setup there. I like that. Let's. Libraries are usually marked with the high background of mRNA levels because of cellular debris. So the question is there a methodology that's implemented on the Singulator to address the issue while preparing single nuclei suspensions?
Yeah, thanks. Great question. So the cartridges have actually 2 filters built into them. For the nuclei cartridges, there's a 150 micron filter followed by a 40 micron filter. The nuclei themselves are in the range of about 8 to maybe 15 microns, 20 microns or so, depending on the tissue of the organism. And so all of the large debris is more or less filtered out. There's some that gets through the filters, but not very much. The small debris can get through the filters, and it really depends on the tissue type for how much of that gets generated. So oftentimes our customers are using a cell sorter or a QUIC gradient centrifugation protocol that we have, manual strainers, or just repeated spin downs to dilute out essentially the debris. Those are all methods that people are using to clean things up further if they need to. Some tissue types, you can go directly into your library prep. But for other tissue types, it's important to clean them up further.
Okay. I had a quick follow on that one thought that came into my mind with the really small amount of sample that you need. And I was thinking about it in terms of the theme being spatial. Is there or are you seeing anyone just taking from a tissue a small sample from different locations to kind of get some spatial information about what's in there?
Yeah, we do. I mean, we have people doing micro dissections of brain samples, for example. We have one customer that's working on honeybee. And so he does a micro dissection of the different parts of the honeybee. We have people doing Drosophila larvae, very small pieces. So yeah, people are doing that. And we recommend at least 10 milligrams of tissue, but we have customers that are regularly using 4 or 5 milligram pieces. We've done OCT slices that are too small to even really weigh, and they've all worked.
Wow. Okay. Cool. And let's see, Diego, your colleague there, throwing the questions out for Govind here. Can you further explain the work using ITP in combination with the transcriptomics, and what does the ITP enable in this particular case?
Of course. Thanks, Diego. It's actually quite straightforward. It's for purification. So no different from what you guys are used to, Diego, and what Purigene does as an example. So it was for nucleic acid purification from the cell debris.
Okay. And then message coming out from John. All right, mistook that one for a question. Okay, let me check my notes here. If anyone else has questions they'd like to ask, go ahead and put them up in the chat room there. And let me think. The one thing I was thinking about the coaxial approach, it really spurred a thought in my mind with some of the early work that was done by Gary Van Berkel and his group down at Oak Ridge with the, they had the kind of coaxial tubes with fluid going in and going back out for doing mass spec analysis. And I think, was it Advion eventually licensed that to do kind of surface sampling. I think it was sort of what led to my question about the size, and I can't remember how big theirs was. I think yours has definitely got better spatial resolution, but they had gone down the path of just having a simpler tubing configuration. I don't know if you'd seen some of that as a part of the stuff you were working on before, or familiar with it, or if it had any impact.
I have actually. I've seen this work, and there's a few more different works coming out of. I think one is from Utah. The name is just being elusive to me at this hour.
Yeah, okay,
but,
okay.
There have been a few others. It really depends on the application. Not everyone needs to push resolution.
Yeah,
actually,
yeah.
I would say one of the bigger criticisms we had for some types of work that we were doing, or certain applications, was, you're perhaps being too small. You need it to be much larger to have an optimal sample of the surface, depending on if you are patterning receptors on a surface, and depending on what type of application we were doing. We were actually told from the application space people that you should go to almost 1 millimeter length scales.
Oh, wow. Okay. Yeah.
So it was, but since it's a sampling, it's on a scanning stage, it was easy to rasterize and come up with ways to do that. But also the concept of flow focusing is not limited to these scales. You could go much larger.
Yeah.
Of course, with certain caveats of, using more reagents, but then you do better recirculation to be able to have a trade-off between the two.
Yeah, I remember seeing huge arguments in the mass spec community about similar issue in terms of resolution. I think they came back around how fine of the resolution could be on the MALDI imaging systems, and I think they eventually came back to the point where it was just too small as well. So it's kind of a nice circle to close in some ways, right? Yeah.
Some of the arguments actually we used initially also, which was in favor for us, was the work of the tissue microarrays.
Yeah.
So back in the day when tissue microarrays were developed, actually the genesis was in Bern here in Switzerland for the pharma as a test platform. It was not really accepted very well, saying that you might be losing a lot of information. But over the years, it got accepted, and actually it's now routine to use tissue microarrays. So that says that you don't necessarily need to have the entire section, but you could use a fraction of it and be able to more or less have similar performance in terms of representative of the original sample.
Yeah. Okay. See, we still got some time. So maybe I see Diego put the question up for John. John answered. Maybe you want to comment on the question on how sensitive the nuclei isolation protocols are to the tissue stiffness, and how does that compare to expected or natural stiffness variations in nature? So what if a kidney tissue is stiffer than you expect?
Sure.
Maybe,
yeah, the protocols are really broad. We find that the protocol optima are very flat, they work well for really broad ranges of tissue characteristics. So we have actually we have three basic protocols on the system. The normal one, the standard one, covers 75–80% of tissue types. Then we have an extended protocol for things that are really mechanically hard, like the prostate tumor I mentioned. And then we have one for small particulate samples like Drosophila brain. And then also in the software, there are user selectable parameters that you can change to tweak a protocol. So you can, for example, change the speed at which that grinder mechanism spins. And so if you've got a kidney sample that's maybe a little bit more fibrous than normal, or it's an animal model for fibrotic kidney, you can just turn the speed up or down on that grinder for whatever tissue characteristics you have, and it's very easy to dial it in. That's really the only of all the parameters that we allow users to change. That's really the only one that anybody ever changes. Is the speed of that grinder. And so if you got brain, turn it down. If it's prostate, turn it up.
Yep. And do you have to vary the, I know you mentioned yet enzyme, do those have to vary much from tissue type to tissue type? I can imagine that going along, and I assume you guys will include that as, support for researchers depending on what type they're working with?
Yeah, again, for nuclei, it's one reagent for everything, but cells, it is different enzyme cocktails. So we provide kits for a variety of different tissue types. And we find that they're generally extensible. If we have a mouse kidney kit, it works fine on human. But the system is also open so that if you have an enzyme cocktail that you really like for a manual process on your particular tissue, you can use that on the Singulator, and we can help you port that protocol onto the Singulator. Usually it's very easy to do that, and usually the protocols end up taking around half the time from your manual prep, if not shorter.
Yeah, okay. And then, see, I got one more that I knew I just had to find it in my notes here. For Govind, you mentioned you were going and you're starting to work with people in a clinician type setting. Once you get to that stage, always becomes interesting from my experience. You get into the customer labs and you learn which things that you anticipated aren't exactly as anticipated. Have there been some interesting things that you can share that were, as you moved into this phase of trying these things out, that were potentially surprising? I'm thinking about this from the perspective of people who may be trying to develop tech to get through that phase without being overly surprised.
Yeah, I think each of us have to experience that firsthand for the best experience. So maybe there
You're
was
kind of
one or
missing
two
out if you
things.
don't, right?
Yes. So I could think of maybe one or two things which is linked to what I spoke earlier today. One was many of the pathologists actually told us that, for particularly when you're looking at immunohistochemistry, think of having much larger dimensions. Because you might miss out, even if you can do multiplexing, you might miss out a bit. And if you have to do certain type of scoring, they will feel much more comfortable if you're having at least in a millimeter length scale, not in the micrometer length scale. That was one aspect which came up. It was counterintuitive to what we thought. We thought the better the checkered pattern we can have, the more fascinated they would be.
Yeah.
They were fascinated, they were intrigued, but they were not impressed.
Yeah.
They wanted something which they can really use on the ground. Now, second was one aspect about fish assays, which actually was very surprising to us, was there was such a huge variation in the protocols from lab to lab, as well as from how each of them have do implement it. There is a lot of history of the lab, which is a function of what is the current assay.
Yeah.
Like maybe some technician at some time 5 years back developed for this particular tissue, a 12-hour incubation is good. It's always 12-hour incubation. It's not 11, it's not 13. When you ask them why, there is no specific reason beyond that this is the protocol and this is what I'm going to use.
Yeah.
So we started thinking about rational design a bit more, introducing um, specific reasoning for design of an assay. There was quite some resistance, but they were open to it eventually.
Yeah.
And
Yeah.
we started working with some of the groups to be able to redesign that. And in some cases, what they had was actually the most optimal, strange enough, without realizing um, it was optimal, they were using the optimal. And in
Yeah.
some cases, we actually could shave off 2, 3, 4 hours from their incubation times or overall their process in their workflow. So I think sometimes it's easy to criticize not doing rational designs, but experience brings in interesting optimas. And other times, actually, you could use a back of the envelope calculation to optimize what currently is used in the clinic.
Yeah. Yeah. No, we used to always call it running into the tribal knowledge wall, right? Kind of
Yeah,
things that have
exactly.
been passed
Exactly.
down for generations. Nobody knows exactly why that parameter was in space,
Yes.
and
But it's
it
good
takes
to
a
question
while
it.
to
It's good.
convince them.
Absolutely. But it's good to question it, and it's
Yeah.
good to sometimes accept it humbly that what it is works, and
Yeah.
other times you could try to optimize it. So
Yeah.
it could work. Unless you ask these questions, there is no opportunity to try to get them better.
Think about that along the lines of your protein quantification data. It's kind of setting up the calibration. I wasn't quite sure, is that one where you'd be able to do a little bit like you had shown a little bit earlier in the talk, to do a bit of a time course measurement, just a real-time time course measurement of the flow across, and watch the thing growing, then just map out a real-time kinetics measurement? Is that something that
Absolutely.
would be feasible?
Exactly.
Okay.
Yeah, that's exactly where we were going with that work.
Yeah, and that would be one where they would completely not be thinking about it that way.
Yeah,
Right?
absolutely. Because immunohistochemistry has almost never been quantitative.
Yeah.
People talk about digital pathology, they talk about digitizing it, but still that's an
Oh,
endpoint measurement. And that's what they failed. So when we spoke to a lot of people in the pathology community, they said, oh, we are doing completely quantitative stuff. We are scanning the images, or we are scanning the tissue sections, and we're working with images. So that's really as quantitative as it can get.
yeah.
But the point is the quantitation, that yes, it certainly improves quantitation, or let's say it reduces errors of interpretation between one pathologist and the other to a certain extent, but it doesn't add the quantitation aspect to it, because you're really still looking at the endpoint data.
Okay, super. I think it's a good place. So guys, I'm gonna wrap up my questions here. I don't see any more in Again, I want to thank John and Govind for being with us tonight. And at this point, I will pass it back over to Diego.
Yeah, it's actually going to be me this time.
Oh, back to Thomas. Okay, there we go.
So I'd like to thank Kaigala, John, and Don again. That was a great discussion. Really insightful talks. So the next section of our event is our job posting or jobs board section. We did not receive any job postings to this event beyond the ones that John mentioned at the end of his talk. So please reach out to John if you're interested in those positions. Based on personal experience, though, I know a lot of companies are hiring right now. So if you're looking for a job or if your company is hiring, I encourage you to join our jobs board. It's a Google group, so anybody can send a message advertising that they have a job available. It's about 400 people on the list, so it's a pretty good audience.
Yeah, I can agree with Thomas. From what I've seen from my spot, there are a lot of openings right now.
So I encourage you. I just posted the link in the chat, or I'm about to post the link in the chat. There it is. So just click on that and you can join. So next after that is our networking session. So I hope that you can all stick around for our networking session. We will momentarily open the breakout rooms. And last but not least, thank you all for joining us.
Okay.
Hope to see you at the next one.
Yep. Thanks, Thomas.
Great. So I've just opened up the breakout rooms, and people who had already selected the breakout rooms should already be in there. But I would also be assigning breakout rooms to people. So let's see. Okay.